Hello and welcome to Go Cognitive. This is our third and final segment with Dr. Kelleher from the University of Idaho. And we're going to talk a little bit more about olfaction and research design. So thank you and welcome, Doctor.
Thank you.
My first question is, why do you choose to use mice in your studies?
Well, certainly mice, unlike some other species, utilize olfaction for almost every aspect of their life, whether it be mate attraction, whether it be aggressive behavior, certainly finding food. And so they're macrosmotic animals that are utilizing chemical senses, in particular smell, in order to influence almost every aspect of their life. So by using that type of animal as a model, we can then understand a little bit about what's going on in the brain in terms of receiving these sensory cues and processing them, and what ends up eliciting in our interest social behaviors. So while me and you might use vision and audition, and mice are primarily going to use olfaction, eventually they're going to end up triggering the same mechanisms in the brain though. And so we really want to understand how that occurs.
Can you talk a little bit more about the individual systems within the mice's olfaction?
So in their nasal cavity, we talk about them having as many as 6 distinct subsystems or olfactory organs in reality. But even more than that, each of those systems has subsystems associated with them as well. So not all olfactory neurons are made alike, and certainly not all vomeronasal neurons are made alike. So I mentioned that and mice will have a main olfactory system, which classical olfactory receptor neurons that project to the olfactory bulb and are brought to the brain from there. They also have a system called the vomeronasal system, which is a distinct organ in the nasal cavity. It's located at the base of the nasal septum, and it brings in generally mucus into this organ. It then binds receptors located in that organ, and those are sent to a distinct area in the olfactory bulb called the accessory olfactory bulb. So those neurons project up to the accessory olfactory bulb and then are passed along to other areas of the brain, usually different nuclei located within the amygdala, some direct projections to the hypothalamus. So even in those two systems, main system and vomeronasal system, there's subsystems. For instance, in the vomeronasal system of most species, or most species with a well-developed vomeronasal organ, they have a distinct class of neurons that are located apically in the epithelium, and a distinct class are located basally in the epithelium. And these two regions, the apical and basal, are still neurons that are receptor neurons detecting chemical cues, but they probably detect different classes of cues, and they send them to different areas of the olfactory bulb, an apical or rostral region and a caudal region of the accessory olfactory bulb. And so there's these distinct subsystems there. In the main olfactory system, they have different neurons that are located within that epithelium, but they have different transduction pathways and usually have different projection areas in the olfactory bulb. And so we can classify these as olfactory subsystems, differences in the signal transduction and the projections, where they go. They can be thought of as different subsystems, and likely are going to be involved in different aspects of social behavior, and possibly integrating different aspects of behavior as well.
So you've described the different systems in mice. Can you compare that to the human olfactory system?
So certainly in humans we don't have all the same systems as mice. For instance, the vomeronasal system in humans is most likely a vestigial system. That we have a lumen in that general area, but whether or not there's functional olfactory neurons, it's unlikely. One of the reasons why we know this is because there's particular signal transduction components. There's this transient receptor potential channel, or a trip channel as we call it, that's specific for the vomeronasal neurons. Mice have it, vast majority of mammals have it, humans and primates don't have it. It's no longer a functional gene, it's called a pseudogene. So some point during evolution. Stop codon that mutated. A stop codon was placed somewhere in the region in which it produces an actual protein or transcribes to a protein, and it was lost, and humans kept on evolving regardless. So certainly there's this difference in that mouse has a vomeronasal system and we don't have a vomeronasal system. But the question then becomes, what do we learn from that? One thing that we learn is certainly we're utilizing similar type cues, but detecting them now through the main olfactory system. We no longer need this vomeronasal system. So odors that can be detected by the vomeronasal system oftentimes can also be detected by the main olfactory system as well. And how they're processed just seems to be a little bit different. One example is the MHC complex that we had talked about before. In rodents, it's absolutely detected by the vomeronasal system. It's detected by basal cells in the vomeronasal organ, and those cues are processed in the brain, in the accessory olfactory bulb first, and then in the brain as being able to recognize individuality based on those cues. However, we also know that the same exact MHC molecules can also be detected by olfactory neurons in the main olfactory epithelium and are likely also communicating individuality. And so just because we don't have a vomeronasal system doesn't mean we can't detect those cues and process them in a similar way that the vomeronasal system would have processed. Of course, there's going to be other examples where we've lost that ability to sense that chemical cue, and it's just the fact that we don't need to detect that chemical cue anymore. It's just not as important to us anymore. We've utilized now other sensory systems in order to communicate that information that these mice and rodents need to have. And in that case, we're still learning about how these cues are being processed in those nuclei, in those brain areas. And it's going to be the same behavioral mechanisms that are going to be elicited by rodents as they would be in humans as well. The same, it's just the trigger now is different. So instead of having an olfaction cue as a trigger, maybe we're using visual cues as a trigger or auditory cues as a trigger. But the same neural mechanism underlying that social behavior is going to occur. And so we need to be able to study that neural mechanism in an animal which we know we can elicit the behavior, and we know exactly the type of cues which elicit that behavior. And from there, we can really understand what that neural mechanism is. And then to link it to humans later, we have to identify what other cues humans use to elicit that type of mechanism or social behavior.
Can you describe some of the research designs and research tools that you use to gather this information?
My lab, we use 3 different types of techniques generally. We have a behavioral component to the lab, and where we look at animal behavior and how olfactory cues are mediating different aspects of social behavior. We have a part of the lab in which we look at neurohistology, and we actually look at the brain, and we can actually image different areas of the brain, in essence utilizing different markers for activity or different neurochemistry in the brain that is being utilized. And then we're starting to do some work using electrophysiology, where we can actually measure electrical recordings, generally in the epithelium of animals right now, and what type of cues are eliciting what type of signals in which particular pieces of the olfactory system. And in many animals, and quite possibly in humans as well, there's an aspect of social behavior that is mediated by olfaction. It's called food preference, socially transmitted food preference. And what we see is that mice, and this happens in dogs and other species, that the animals are able to form preferences for a particular food type based on what's on the breath of another animal. And so if this animal over here had been eating something flavored with cocoa leaves or with cherries or what have you, if another animal was to come over and smell the breath of that animal, they would form a preference for that food, most likely thinking that, okay, this is a good food to eat. But what's really interesting about this is it's a combination of different sensory cues when you think about it. You need the odor from the food, but you also need the breath of the animal in order to elicit this preference. The animal won't form a preference if they just come up and smell it on the body of the animal, or if the animal is anesthetized and it smells it on the rear end of the animal. They won't form a preference to it. They need the combination of breath, which would be the social cue, and then the food odor, which would be the food odor cue. And so we're doing a number of studies utilizing this paradigm and trying to understand what it is about the breath. What are those breath cues that are activating this social transmission of food preference? And from there, hopefully we can understand, if we know exactly what those cues are, we can understand what system they're working through, and we're going to understand how they integrate in different areas of the brain, whether it's peripherally they integrate at the level of the receptor, maybe they integrate at the level of the olfactory bulb, but somewhere along in the brain, there needs to be an integration of these food odors and these social cues. And understanding where that happens is going to lead us to understand how social communication can occur.
Can you tell me what other research projects you're working on other than that dealing with food preference?
Absolutely. We actually have a fascinating project going on right now. We think of mice should be, in general, running away, show defensive behaviors in response to predator odors. However, there's an instance in where a mouse can be infected by a particular parasite called Toxoplasma gondii. Toxoplasma gondii we know is causing toxoplasmosis. It's a protozoan parasite that can infect just about any warm-blooded animal. In fact, it's found in humans as well. And it enters into the central nervous system. It'll get into the brain and form cysts in the brain, just latent cysts. And in humans, it's thought to be completely benign, although we're starting to question that right now. So the interesting thing about this particular parasite, it's found in cats, but it's only sexually reproductive in cat gut. And so it only goes through its complete life cycle when it enters into the cat, is then excreted from the cat, and then the cat eventually consumes it again. And so there's a hypothesis, part of the host manipulation hypothesis, in which it's believed that infection of a rodent or a bird might elicit differences in behavior in that animal that would make them more susceptible to predation. And so the thought is this parasite can actually get into the mouse and then elicit a certain behavior in the mouse that's going to make this animal get eaten by a cat and complete the life cycle of that parasite. Kind of scary thought, a parasite in the brain controlling behavior, but it's not out of the realm of possibility. What we think is happening here is it's actually manipulating the olfactory system. There are a number of researchers who've been studying how it influences behavior. We're more interested in how it might be actually manipulating different parts of the olfactory system, the chemosensory pathway, either by blocking the ability of urine cues from a cat or cues that would normally elicit aversive behavior in a rodent, either by blocking those cues specifically or altering one part of the pathway in which normally defensive signals are being processed and elicit defensive behaviors, possibly altering that part of the pathway so now that cue is actually being sent through what would be considered the reproductive pathway, and where the animal would now be attracted to that odor.
You talked about behavioral effects of being infested with this parasite. What are the effects on a human?
So that's interesting. In general, it's thought to be just a latent infection. More and more people are thinking that maybe the cysts in themselves could be having effects. Personality disorders, I suspect possibly could be influenced by whether or not this parasite's having some sort of an impact, especially if it's in an area of the brain, that is important for personality or emotion. In the case of humans, whether or not those links are particular to chemical senses, we don't know, but certainly if we can understand what parts of the brain that are influencing the behavior in these rodents, and so allowing the cues now that are outside the body to be manipulated as they enter the body and elicit a change in behavior, if we can understand where that's happening in the brain, we could quite possibly link it then to the types of behavioral defects that might be seen in people as well. It's going to be the similar areas of the brain again, Maybe it's different cues, maybe it's visual cues or other social aspects or memories that enter into those brain areas. But they will be most likely similar mechanisms underlying alterations in behavior.
I want to thank you, Dr. Kelleher, for doing this interview. I learned a lot about the fascinating system of olfaction.
Well, thank you very much. It was a lot of fun.